STB 3182. Lecture 3 Subgroup 4. Biosafety in Contamination Handling. Group Members. Biosafety is the prevention steps to determine and manage the potential risks that harm living organisms and ecosystem. The importance is to ensure proper handling of infectious organisms and hazardous biological materials. Keeps personnel safely in lab and also protects outsider comes in contact. Biosafety levels are classified into 4 levels: BSL1, BSL2, BSL3, and BSL4. All biosafety levels refer to microbes that cause diseases to humans and animals. BSL2 has a risk to the environment and cause serious infection but has effective treatments. Conversely, BSL3 does not transmit among humans while BSL4 is transmitted among humans where no effective treatments. How to handle laboratory contamination. 3 main components: lab practices, safety equipment, and facility. Do not return excess reagents or chemicals to their original container. Sterilize the labware before using and after contact with the cells. Use chemical fume hoods to reduce the harmful effects of chemical inhalation and exposure. Wipe the fume hood with 70% ethanol before and after every use. Cabinet laboratory. All infectious agent work is done in a Class 3 biosafety cabinet with careful protocols. Suit. Laboratory workers wear full-body, air-supplied suits. Before that, workers go through few procedures to disinfect them. Exhaust air from the Class 3 biological safety cabinet must pass through 2 HEPA filters before being released outdoors. The cabinet always operated at negative pressure. A non-recirculating ventilating system is important. Steam autoclaving is the preferred method for all decontamination processes. Materials for disposal should place in containers according to color-coded autoclavable plastic bags to autoclave. Contaminated hypodermic needles, scalpels, placed in a sharp bin and treated as infectious. Check the samples regularly for any contamination occurs. Label all the flasks, culture samples, or potentially infectious samples properly to minimize the spreading of infectious agents. Use an excellent microbiological technique. Personnel working with biological agents must be aware of hazards and trained to properly handle and dispose of contaminated materials. Laboratory-specific manual always prepared and accessible. Wear a face mask, gloves, lab coat, and covered shoes to protect chemical splashes, exposure to biological. Wear lab coats only inside the labs and clean them often to prevent the contamination spread outside the lab. The International Biohazard Symbol and Biosafety Level displayed on lab doors. For directed airflow, air gaps under doors are suitable. Solvents and radioactive materials should be handled and stored carefully, hence need more space. Safety systems should cover fire, electrical, showers and eyewash emergency. First aid areas are easily accessible. Windows should be opened and fitted with arthropod-proof screens. Laboratory biosafety is crucial because it helps to protect us from potential contamination.